Okay, this is the kidneys. So let's start with a little bit of context as to where we are. I'm going to draw a miniature kidney down here. Look a little bit like a kidney bean, which is no coincidence. It's got two sections. We've got sort of the inside and the outside. This is fed by blood supplies called the renal artery, and the blood comes out of the renal vein. So if you hear the word renal, see the word renal, it means kidney, much the same way that pulmonary means lung and cardiac means heart. The outside part of the kidney here is called the cortex, and the inside part is called the medulla. And the bit that we're looking at in this section is going to be taking place on the boundary. So this is my Bowman's capsule and my proximal convoluted tubule. The loop of Henle goes into the medulla, then we have the distal convoluted tubule, and then the collecting duct, which I suppose is going to connect up with my ureter. The ureter goes to the bladder, which connects to the urethra, through which you piss. Okay, so this is the thing that we're going to be looking at. One of these things is called a nephron. So let's put that up here. Right, now I'm going to do some drawing. I'm going to get it sped up so you don't have to take the time to watch me draw it nice and slowly, and then we're going to label it and fill it all in. Okay, so let's start putting some notes on this thing. So this whole thing in black we can refer as a nephron, and it's surrounded by blood supplies, by capillaries. Let's start with that over here. So we can see we've got a wide end and a narrow end. This is the entry end from the renal artery, and this is going along. The renal vein is going to be at the other end down here. So this is a bit like putting your thumb on the end of a hose pipe. You're going to build up high, what we call hydrostatic pressure. Going to be a high pressure in the liquid, which is in here, which is basically going to force stuff out in that process. Because it's under pressure, we call it ultrafiltration. So filtration under pressure. So we call this wide end, the entry end from the renal artery, we call that the afferent arteriole. Before I put too many notes in the way, I'm going to just label the difference between my cortex and the medulla. Okay, so everything on the bottom half here, so the loop of Henle and the collecting duct, when we've labeled it, are going to be in the medulla. And then everything else, the Bowman's capsule, the proximal convoluted tubule, the distal convoluted tubule, are going to be in the cortex. So the afferent arteriole, it comes from the renal artery. Blood is going to go in this way. We can maybe remind ourselves which one's which. The afferent comes first. The afferent is the wide one, so we can say that it's larger. And the one coming out the other end is called the efferent arteriole. This big ball of capillaries on the inside here has got a name. It's called the glomerulus. And the glomerulus is inside this cup-shaped thing, which is called the Bowman's capsule. Okay, everything after this socket-shaped thing up to this big U-shaped thing, we call the proximal. Proxima is like the first, the closest to the start, the proximal convoluted tubule. And we can call this, always write it in full first time, PCT is an accepted shorthand. Okay, so proximal convoluted tubule, everything here is convoluted. It's bent, it's curved, it's shaped in a funny way. This could look slightly different. The exam might be bent on itself, it might be more vertical. However, the loop of Henle and the collecting duct will be the only things in the medulla, that's for sure. I'm going to put some notes on this. So what do we need to know about it? Well, all the stuff from we'll call it the dirty blood, gets forced out by ultrafiltration. All the small stuff, so glucose, sodium ions, we've got the urea, all the bits and pieces like that are going to be in here, and that was called the filtrate. And we need to reabsorb basically as much of the good stuff as we can. All the glucose, you don't want to get rid of that because you work hard to find food to get the glucose in the first place. So the most of the reabsorption takes place in the proximal convoluted tubule. This includes water. The fine-tuning of your osmoregulation, water regulation, takes place between the loop of Henle, a little bit in the distal convoluted tubule, and the collecting duct. But most of the water is actually reabsorbed from the proximal convoluted tubule. It's doing a lot of this by active transport, so there's lots of mitochondria. And we can say that for large surface area, we have microvilli. I'm just going to write capital S, capital A for surface area. I'm going to be quite tight on space. Okay, so what next? We've gone through the proximal convoluted tubule. This next U-bend type thing is called the loop of Henle. Another capital here, capital for Bowman. They're both named after people, so they have capital letters. We can divide the loop of Henle into two sections. We've got the descending limb and the ascending limb. At the top of the loop of Henle, we then enter the secondary, the distant, or we can call it the proper name is distal convoluted tubule. Again, we can call this by abbreviations and call it the DCT. What do we need to know about it? Well, it's the fine-tuning, it's the reabsorption of sodium, basically. And then the last section is this bit down here, is called the collecting duct. Okay, perfect. Where does this capillary go? Well, this goes off towards the renal vein. And the urine, which is the product of all of this, goes to the ureter. The ureter then goes to the bladder for storing the urine, and then when you want to pee, it comes out of the urethra. Okay, next up. We've covered what the proximal convoluted does, active transport, reabsorption of stuff to get the good stuff back out of the filtrate. The loop of Henle is mostly concerned with osmoregulation. So this is fine-tuning how much water do you need. The collecting duct is also pretty key in the osmoregulation. And we're going to look at, if you need to know about ADH, antidiuretic hormone, then that's going to be mostly impacting the collecting duct, a little bit on the distal convoluted tubule too. Okay, so a very quick overview. We're going to look at each of these sections in turn. We're going to look at ultrafiltration. There's going to be a lesson on that. We're going to look at selective reabsorption, which is going to be what happens in the proximal convoluted tubule. We're going to look at osmoregulation controlled by the loop of Henle and the collecting duct. There's almost no questions actually on the distal convoluted tubule unless it's related to osmoregulation. So each of these is going to be looked at in turn. I think that's pretty much, from a structure, that's all you need to know. In those lessons, I'm going to be talking about these things as if you know them. So that's why I've done a nice overview of the kidney here.